Hello, everyone, and welcome to today's session. My name is Mary Obege-Brins. I'm a Hamilton Robotics employee and will be your moderator today. I'd like to introduce today's speakers. Our first speaker, Dr. Gabriela Boza-Moran, is the Scientific Content Manager at Hamilton Robotics. She has a PhD in molecular and cellular biology from Royal Holloway, University of London, and an executive MBA in Management of Technologies from the Swiss Federal Institute of Technology in Lausanne, Switzerland. Gabriela has more than 10 years of experience in scientific research. Our second speaker, Dr. Federico Sebastiani, is the International Commercial Leader Diagnostics at Hamilton Robotics. He's also responsible for partnerships, business development in Europe, Middle East, Africa, China, and India. Federico has a PhD in molecular and cell biology from Pavia University in Italy and has more than 20 years of business and commercial experience in life science, diagnostics, and biotechnology. Before I hand the mic over to Gabriela, I have two housekeeping items that I would like to share with you. First, this webinar is being recorded. Next, we'd love to hear from you during today's presentation. If you have questions for our speakers, please feel free to send them through the Q&A box at the bottom of your screen. I will be collecting your questions, and both our speakers will be answering them at the end of their sessions. If we don't get to your question during today's webinar, don't worry about that. Please feel free to contact the speakers after this webinar via email. You'll find their email addresses at the end of this presentation. For those of you joining us, welcome. We will now begin our webinar, Automation in the Diagnostics Market, Developing Solutions to Accommodate a Variety of Throughputs in Highly Regulated Environments. I will now hand over to Gabriela, who will start today's presentation.
Thank you, Mary, and good morning, everyone. Thank you for joining us today in this webinar. I would like to start by describing the way this webinar is structured. So we will start with a short overview of Hamilton's business units, and then we will dive into the core of this event, which is divided in two parts. The needs of the end customer in the clinical diagnostic market, and then the needs of the manufacturers of diagnostic kits. In both cases, we will describe how Hamilton can provide solutions for each of those needs. We will end with a case study of one of our collaborations. So let's start. An overview about Hamilton. Hamilton is a private group founded in 1947 by Clark Hamilton in a garage in California. Today, Hamilton has two headquarters, one in Reno, Nevada, and one in Bonaduz, Switzerland, and a total of 5 manufacturing sites, which includes sites in Franklin, Ems, and Romania. Altogether, the company employs more than 2,500 employees around the world, and this includes office in 7 subsidiaries in Europe and in China. Hamilton is divided in 5 independent units: robotics, storage, process analytics, laboratory analytics, and medical. So Hamilton Robotics is specialized in manufacturing of liquid handling instruments, for both for OEM and for direct sales. And this particular webinar is focused on products and services for this particular business unit. Hamilton Storage manufactures ultra-low management systems and small devices for capping and decapping. Hamilton Process Analytics manufactures measurement devices, including sensors for pH, dissolved oxygen, and conductivity. Hamilton Laboratory Analytics manufactures syringes for use in various applications. And finally, Hamilton Medicals manufactures ventilators for people in critical care. So Hamilton Robotics has 4 platforms for automated liquid handling solutions. So we have the ML Prep, the Nimbus, the Star, and the Vantage. In this webinar, we will focus on the Star line, our line of IVD compliant platforms. So after this brief introduction of the company, let's just talk about the customer needs in the diagnostic market. And this includes laboratory personnel, physicians, and patients. So what we have here is a list of 8 of the most common needs of the end customer or the end user. And we will see in detail what are the solutions that we, Hamilton, can provide to cover those needs. So we have higher throughput, faster turnaround times, ease of use, precision, reliability, reproducibility, traceability, safety, ability to control workflows, efficient customer support, and cost efficiency. Let's start with the first 2, high throughput and faster turnaround times. The need for higher throughput is particularly relevant for large-scale hospitals with established testing facilities. Dedicated testing facilities can process thousands of samples per year for NGS analysis or even thousands of tests per day for LC-MS analysis. With such a high number of samples, laboratories and physicians struggle to deliver results within a short time, something that can actually hinder the medical treatment. So one of the ways automation allows for higher sample throughput is by using the 96 probe heads, which allow quick pipetting of entire or partial plates. Our 96 probe head can work with single columns or rows, and it operates with volumes from 1 to 1,000 microliters. Another way to answer to these needs are the way our systems are built. Hamilton systems are built on the principle of modularity and flexibility. Our decks can be customized according to the kit's workflow and or the customer needs. So we have modules for common activities such as shaking, centrifuging, temperature control incubations, amplifications, etc. Our instruments can be upgraded at any moment in order to add new modules or pipetting channels, or also to integrate third-party devices depending on the system. In collaboration with Hamilton Storage, we can also integrate compact or large storage system to our instruments like the one we see here. In order to coordinate activities among the several third-party devices into one automated workflow, we can design solutions that include our H-Motion, which is our small footprint robot for bench integrators. And we see it here. I'm gonna click to see if, I think I have to disable the laser so I can put the video. Second. I hope you can see the video. It's a little bit showing how it works with the H-Motion. And if you can see it, the H-Motion can pick up and transfer plates from our these instruments to another. And this allows for a complete automation of complex workflow. Just gonna give it 2 seconds because I know it's not always easy with webinars to see how it works. Now moving to the third need, ease of use. As you can imagine, these levels of integration and technology can seem very intimidating for some users. The handling of some instruments and workflows can require a significant amount of training, from weeks to months. And we know that the need for expertise is one of the biggest bottlenecks in most laboratories using advanced technologies. And customers are looking for easy-to-use solutions. In order to provide end customers with easy-to-use solutions and easy-to-implement solutions, we have what we call asset-ready workstations. Asset-ready workstations are pre-designed deck layouts that has been already programmed to perform qualified workflows. This means that the end customers already are equipped with all the tools, hardware, and software needed to perform their experiments. So here we show 3 of asset-ready workstations: the Nimbus Presto for nucleic acid extraction, the NGS Star for NGS library preparation, and the mass star for sample preparation for LCMS. In the case of IVD instruments, the systems are always provided to the end customer as a ready workstation. However, during the development phase, the kit manufacturer works with Hamilton to develop partial or complete customized solutions based on our existing systems. Precision, reliability, and reproducibility. The complexity, length, and high sample throughput of clinical workflows results in a high susceptibility to manual processing errors. Even with the availability of experienced qualified staff, inter-operator variability still remains. In a clinical practice, an incorrect result could result in withholding effective therapy or administering unsafe therapy. Automation allows for very precise and reproducible workflows. Hamilton has developed several pipetting technologies to ensure precision, reliability, and reproducibility. The first one is compression-induced O-ring expansion for tip attachment and positioning. I'm gonna use again the laser pen, the laser point here. So if we look at the fair image here from the left, we see the pipette channel on the top and the tip on the bottom. In the second, we see the pipette channel that is entering the tip. And in the third one, we are seeing the same but attachment of the pipette channel in the tip. The key of our technology is actually the locking of the O-ring in the pipetting channel with the annular groove of the Hamilton tip. This system requires no vertical force for tip attachment or tip ejection because it's only based on the automatic compression and relaxation of the O-ring here. This ensures precise tip attachment and positioning and also minimize aerosol contamination, typically seen when tips are ejected with pressure. Next, we have the air displacement technology. Here we see our pipette channel, our tip, and our sample. Our pipetting system is based on air displacement pipetting, comparable to the functionality of handheld pipettes. Air displacement means that the liquid is aspirated into and dispensed from a disposable tip or needle by the movement of a plunger. So between the plunger and the liquid surface there's air. No system liquid of any kind is involved, thus producing less contamination. We also have the anti-droplet control, ADC. This functionality is very useful when working with volatile liquids. Here at the top of the graph, here on the left, we can see what happens in conventional pipettes. The pressure is on the y-axis and then the time here on the x-axis. So the high evaporation pressure of volatile solvents inside the tip causes immediate dripping from the bottom of the tip. So what we see there is that pressure on the tip increases and then there's a drop. Then the pressure increases and there's a drop, increases and there's a drop. Our pipetting channels are equipped with a pressure sensor that detects the pressure increase and immediately compensates by moving the plunger up. This way, the liquid remains in the tip. Based on the sensor on the pipetting channel, we have also developed 3 other pipetting functionalities. Here we have the liquid level detection, which is used to detect liquids and thus the exact position of the top of the sample, and can also differentiate between conductive and non-conductive liquids. In this video, again, I have to remove the laser pond. Trying to remove the laser. All right, sorry for that. So in this video, we see how the conductive liquid can be detected in the sample. This could be, for example, the case of vitamin D extraction with the N-hexane out of human serum, where vitamin D is in the upper non-conductive organic layer, and we remove the lower conductive layer to get rid of it. So just to be safe, I'm gonna keep the laser out. Our next technology is monitor air displacement. It's an aspiration monitoring feature. It measures the pressure within the pipetting channel in real time during the aspiration process, and this allows us to detect clots or empty wells. We also have the TADM, the total aspiration and dispense monitoring. And this is like the big sister of the MAT, or the monitor air displacement. It measures the pressure within the pipetting channel in real time as well, during aspiration, but as well during dispensing. And it compares it to a pre-established range. So both the MAT and the TADM are real-time monitoring. Sample and reagent stability also plays a critical role in the quality control assurance process. Hamilton's automated platforms are equipped with specialized models for temperature control. Here we'll show some examples. So here we have, and first is a PCR comfort lids. It sits on the top of PCR plates and safely protect the samples in the wells from evaporation and contamination during thermocycling or an ODTC. Our comfort lids are single use and are packaged in blisters. We offer several tools that can be easily integrated on our platforms to ensure the stability of compounds stored in various formats. And here we can see, for example, a cooling carrier, which can keep samples sitting on the top cool for hours. This is because the carrier is connected to a chiller. We also have a heating cooling module, which heats and cools SBE slabware from 4 to 95 degrees. This module is usually used for incubations. Our next customer need is traceability. Being able to trace samples during the whole workflow has become an imperative, particularly in regulated markets where a full audit trail is necessary. Hamilton Starlight can be integrated with handheld barcode scanners or equipped with our autoload. The autoload is a device enabling automatic loading of carriers onto the star deck with part of the barcode reading. So hope you can see the video. Let's get it started. Great. As you can see here, it can read barcodes in plates and in tubes, and it has a sensor that identifies if tubes are absent. The scanner in this video reads 1D codes, but we also have a scanner that reads 2D codes. I'm gonna stop the video. We also have the Easy Code, a carrier equipped with a camera that is able to detect 2D codes plates at the bottom of the tubes, which is very common in biobanking activities. So this is the bottom of the tubes, and here's a camera. Our software is equipped with a scheduler, which allows optimization of the workflows. Here at the top, I am showing the scheduler from Venus, the software in a Starline. This is particularly useful for long workflows that need to be replicated from different plates using the same resources, like in the case of ELISAs. So here's a fictional example of that. On the top and color-coded, we see the step of the workflow, which correspond to the diagram shown on the top of the software. We see, for example, here, the loading, transport, washing, transport, pipetting, addition of reagent, incubation, etc. Now, this protocol needs to be done for 4 different plates, but because they all use the same resources, we need to find a way to optimize that. Here we see plate 1, 2, 3, 4. Here on the bottom, we have the resources, which for this protocol are a heater shaker, the liquid handler, a reader, and a washer. So the scheduler allows to see when each resource is used and when it is available. And this allows optimization of the whole workflow. Notice, for example, that when I add 2 more plates, you see here, now we have 6 plates. The schedule shows me that because all the resources are occupied with the 4 first plates, I have to wait 70 minutes to start processing the 5th plate. Safety, ability to control workflows. So part of the quality control assurance in regulated markets is dependent on the user access control and defined protocol for how to handle an error. The access to the Hamilton software controlling the workflow can be defined by assigning different levels of authorization to the users. So the Venus software has 5 levels of authorization. And if we see, for example, for the lab operator number 1 may only run methods, what the lab operator number 2, he can also move elements in the deck. So there are different authorization levels. Our software also have a protocol for setting up error handling, where we can specify what to do in each step. On the top, we can see the possible errors that can happen, and on the bottom, we see the actions that we can take. So for example, here in the step of the aspiration with a 1000 microliter channel pipette, we select that if there is an error in the liquid level cannot be found, then we should repeat the step at the container. And then we should also flag the error. And we can do this for every step in our workflows. Efficient customer support. So customers working in regulated markets, particularly in hospitals, cannot afford to wait days to receive support for their instruments. And they certainly cannot afford to have long instrument downtimes. So apart from the on-site support, Hamilton has a collection of tools for immediate remote support. And this includes, and I go from top to bottom, support classes, which allows the expert person off-site to see what the person on-site sees. LogMe in Rescue to remotely access the instrument, GMX Box as an alternative data transfer, customer support, which is basically a knowledge-based center. And I intend to skip the StarWatch because I want to talk a little bit more about it. And there's a little picture here to depict it. The StarWatch is a software that remotely monitors the condition of our instruments and notifies our field service engineering team when a preventive intervention should take place. This ensures on-time intervention and critically reduces the possibility of errors and downtime of the instrument. So if we start here, so we see that the customer instrument is constantly sending encrypted and compressed data about the performance of the instrument to our StarWatch expert service. This is done by a MST protocol. When there is a pattern predicting that there is a failure observed, then there's an email sent to our backup office who can then schedule a preventive visit from our field service engineer. As I said before, this preventive action can dramatically diminish the downtime of the instrument. So on a final need on the side of the customer is cost efficiency. And is this, as in any other case, so customers are looking for solutions that provide the best value at the lowest cost. So high quality automation solutions like ours increase cost efficiency in 3 different ways. So first is frees up time for more valuable tasks. If you assume that an average person works 8 hours a day, so that would be around 232 days a year, and depending on the day-to-day activities of the person, automation can free up at least 60% of that time. That means that automation is saving that person 139 days per year that he or she can dedicate to other higher value activities. Automation also allows for miniaturization. So let's assume a lab that runs 384 qPCR samples a day. This would translate into around 100,000 samples a year. Now, the cost of running a qPCR sample in a final volume of 20 microliters is around $1, bringing the total annual cost to $100,000. With automation, the sample can run in a final volume of 10, so half of it, effectively saving $50,000 per year. Automation also decreases the need to repeat experiments due to manual error handling. So if I go back to the example of the lab running 100,000 samples a year, and we assume that 10% of the samples no longer need to be repeated, then automation effectively saves at least $10,000 per year. So I'm using dollars as a reference for everyone. And with this, I would like to now give the microphone to Federico.
Thank you, Gabby. And good afternoon to everybody. So let's Okay. Let me see. Just a second. Okay. Can you see the screen in full screen? Okay, now. So now, thank you, Gabby, for the presentation, the first part. Now we will go through the needs of the manufacturer of the diagnostic kits. So let's start with this five section of the second part. So starting from the customized solutions. Why? Because we, as Gabby already said, we have several number of assay-ready workstation. So in general, when we are approached by a kit provider, or we approach a kit provider, we start a conversation from an assay-ready workstation. Why? Because this is a system that is already ready for a particular application that could be in, for example, library preparation for NGS, or qPCR setup, or nucleic acid extraction, or others. Here you can see a couple of examples. The first one is the MASTAR, developed for Chrome systems. So it's a Starlet-based system where we developed the application for sample pretreatment for the LCMS, and Chrome systems provide the kits. And the second one is another example called SENTiNAT 200. This is a sample to results system where the customer can perform on the deck nucleic acid extraction, qPCR setup, and also the qPCR run. Now let's go how we support the project development, because it's important, of course, to identify the system that is important, but we have to develop then the project. So in the first part of the conversation with a kit provider, we touch three principal points: the technical, the application, and the regulatory. From a technical point of view, we investigate what's the best system. If it's a Starlet, if it's a Star, how many channels, in consequence of the throughput, for example, which active modules have to be on deck. This is connected with the application, of course, because depends on the application, we can offer different solutions or customize an assay-ready workstation for the kit provider and end user needs. For example, is related to identify if there are any temperature sensitive steps in the protocol that have to be taken in consideration. And then for last but not least, we discuss the regulatory part. That means we know that we are in the transition phase from the IVDD to the IVD-R, so from the IVD directive to the IVD regulation, and several things are changing. So in this case, also our QM team is involved in the discussion. For the initial discussion, these are the people that you can contact, or you can be contacted by, because this is the International Diagnostics team, where I'm the international commercial leader coordinating this team. And then we have Maria Eugenia, that is managing Iberia as a diagnostic sales specialist, Regina and Claudia for Germany, Switzerland, Austria, and Benelux. And then we have John for GB and Matthew for France. Then when we started the discussion about how we can manage the collaboration with the kit provider, we can provide two different kind of contracts and kind of collaborations. The first one is the so-called reselling. Reselling means that Hamilton is developing the system, the solution for the reseller, and the partner buy the system and resell it to the end user. Then Hamilton provides comprehensive service and maintenance directly to the end user. So it's a collaboration between the two parties, but let's say the first point of contact for the end user is the reseller. In the co-marketing approach, what we can offer is a collaboration together, the Hamilton and the co-marketing partner, but the two parties can sell independently. Hamilton, the platform, and the partner will sell the kit and the sales. Hamilton, again, also in this case, will provide the service, so installation, service, contracts, and maintenance. So in principle, the big difference between the reselling and the co-marketing approach is that in the reselling, we are selling to the partner, and then the partner will resell to the end user. And Hamilton collaborate and support the reseller for the service part. In the co-marketing, it's a real collaboration between the two companies, and that independently can sell the platform and the kits to the end user. There is another important point that differentiates the reselling and the co-marketing, that is the development of the system, because in the reselling, the system is really customized for the reseller. In the co-marketing, we offer to the partner, to the co-marketing partner, in a say-ready workstation that can be a little bit customized, but not in a way as in the reselling collaboration. Let's go now talking about how we support during the application for tenders, because in the diagnostic market, most of the opportunities are managed by tenders. So it's important during the tender application provide particular documentation. And once that we have the collaboration, so the agreement, the contract signed between the parties, that could be co-marketing or reselling, we can support the kit manufacturer with several documents. For example, the declaration of conformity. This is a document that demonstrates that the system is, for example, IVD compliant. In this case, you can see here that the Starline, Starlet are already IVDR compliant. So we are really up to date in terms of regulation for the certification of the systems. Then we can offer as well the risk management. That could be a general risk management based on the platform, or a dedicated risk management for the system that has been developed for the reseller. And the risk management, the risks are taken in consideration to minimize the problems and to optimize the results of the run. Then we have the technical documentation. This is a general documentation that is provided to explain from a technical point of view what is included into the deck, for example, which are the technical characteristics of the system. And then another important point is then once that we have the contract signed, reselling in particular, then we go through the development phase, and then after the development phase, the system is ready to run the kit of the kit provider, is ready for example, for the regulation IVDD or IVDR. Then it's matter of it's time to certify the system, to register the system. And we know that we are in the transition phase from now to 2022, from an IVD directive to IVD regulation. And we also know that at the moment, the majority of the tests that have been performed are performed in a diagnostic environment are still laboratory developed tests. But it's clear then in May 22, when the IVDR will be mandatory, only 20%, this is an estimation from BSI, only 20% of the laboratory developed tests only 20% of the tests will be laboratory developed tests. A majority will be IVD. So we are already ready for this step because our system, the STAR line, is already IVDR compliant. We know that there are a lot of changes in classification from the IVD directive to the IVD regulation, because the IVD regulation is also risk-based. The liquid handler, like our STAR, for example, are related to the class A because of the low personal risk and low public health risk. But the kits of the kit provider that normally are related to genetic tests or infectious diseases or oncology, for example, are covering other areas like class B or class C or class D. The complete workflow, so the platform together with the software, together with the kit, has to be certified as an IVD-D or IVDR workflow. And in this case, we can support the kit provider with a lot of documentation, as we said, with the declaration of conformity. We can provide also the risk analysis and also the technical documentation and the software compliance, that is one of the most important, and usability compliance, and together with the declaration of conformity for IVD-D consumables. And then in terms of responsibility, this is an example of the responsibility how can be shared. So Hamilton is responsible for the instrument, to develop the instrument in terms of hardware, and also the software that is managing the system, software that is as a system IVD compliant. And then is the responsibility of the kit provider to put together the kit and sometimes the software for the analysis to provide to the end user an IVD-D or IVDR solution. We have also a quite extensive experience in monitoring and reporting the post-sales performance of our instrumentation. So this is another key point in the IVDR in comparison to the IVD-D. So we can support also the kit provider and consequently the end user with the analysis and the reports of the post-sales performance. Is the post-market surveillance. So let me conclude with an example of a project with SENTiNAT Diagnostics and Untitled. This is I want to present this project because it's an example of collaboration between three companies. So Hamilton, SENTiNAT Diagnostics, and Untitled. And the three companies are working together to provide to the end user a complete solution, sample to result, where on the deck and on the Starlet is performed the nucleic acid extraction and the RT-PCR setup with the Sentinel kits, and then is performed on deck also the real time on the two small PCR machines. And for last but not least, the analysis of the qPCR cure is performed by the Untitled software, the FastFinder software. And in addition, Untitled is able to provide a software that can connect through the LIMS all these steps and with LIMS. This means that, for example, we can start from a work list coming from the LIMS, providing the list of the samples and the methods to the system, to the machine, and then the instrument can provide all the steps till the step of the analysis step then is provided again by Ugentech, and then we can close the cycle. So let me conclude with a statement of the Executive Vice President at SENTiNAT Diagnostics that choose Hamilton as a collaborator for the automation of their kits because of the reliability of the systems, of the instrumentation, but also because of the reliability and experience and expertise of the development team. Thank you to everybody, and now I think that we can open the Q&A session.
Thank you so much, Federico. Both Gabi and Federico, we have 5 questions in the panel. We have 2 questions from Tahar Ben-Al-Knoum. The first one is, which technologies, apart from the STAR, could be used for preparation of PCR plates, post-PCR, quantification or normalization? And the second one from Tahar again is, do we offer semi-automatic punchers equivalent to BSD 600? And then we have a third question from an anonymous attendee. What do we think is the next big trend in lab automation, and how will that affect Hamilton? The fourth question is from Mohammed Rafiq. Can we please explain systems developed for COVID-19 test manufacturers? And the fifth question is also anonymous. How do we see the label-free detection is impacting both immune assay development and the existing immune assay market?
Okay, we can start from the first. Maybe
I'll repeat the question again.
Only the first. Only the first.
All right. The first question is, which technologies, apart from the STAR, could be used for preparation of PCR plates, post-PCR, quantification, or normalization?
Yeah. In general, all our instrumentation, so starting from Nimbus to Starline and Vantage, all these systems are able to manage PCR plates, to perform nucleic acid extraction, to perform library preparation. So the systems are, in terms of application, are more or less equivalent. Depends on the throughput that is different from each system, and also sometimes the flexibility and the possibility to, let's say, to have more modules on the deck, because, for example, the Vantage system is a system of 2 matrices. Of course, it's possible to have on deck much more modules, and it's a system also that has a cabinet. And in this case, here in this picture, it's not shown, but the Vantage can have a cabinet below. So can of course manage more samples, let's say, or in some cases a more complex workflow. But in principle, all our systems are able to perform any application. ML Prep is very new. We just launched the system. In this case, of course, because of the deck, of the size of the deck, the footprint of the system, there are some limitations in terms of throughput and possibility to manage complex workflow.
Okay. The second question is also from Tahar. Do we offer semi-automatic punchers equivalent to BSD 600?
Yes, we offer not semi-automatic. We offer a complete automation for punching. It's called the Easy Punch. It's a system that can perform punching on cards, different size, from 1.2 millimeter to 6 millimeter. And so it really is really flexible system. Of course, today we are not able to we are not able to talk about all our solutions, but you are invited to contact myself or Gabby to have more information. We can redirect you to the local diagnostic sales specialist or local territory manager. But our Easy Punch is a system that is very well known in the market, and is an automated system for punching cards.
Okay, thank you. Question number 3 is anonymous. What do you think is the next big trend in lab automation, and how will that affect Hamilton?
This is a very good question. Of course, everybody would like to have the answer, but the big trend in lab automation, you know, could be the miniaturization, for example, of the assays in terms of volume, because everybody would like to save reagents and consequently money and the cost of let's say of the reagents. And this will affect Hamilton, yes, because of course we have to follow the trend, but we are moving in the same direction. So we are already going in the direction to reduce the volume that we can pipette. And this is based on new technologies, and is based also on the fact that the systems are very reliable and very precise.
Okay. Question number 4 comes from Mohammed Rafiq. Can you please explain systems developed for COVID-19 test manufacturers?
Yes, unfortunately, we know that COVID affected everybody a few months ago already. So when around, if I remember well, at end of February, the situation was clearly dramatic. Most of the kit providers kit manufacturers started to manufacture kits, nucleic acid extraction in principle, but of course kits for COVID testing based on real-time PCR. And we were able to develop 2 systems. One is called MagX Starlet, and is a system for nucleic acid extraction. Is a system with a 96 mph, so it's very system for medium throughput, let's say. And then we also developed the MagX Star, so based on the Star, not anymore on the Starlet. So now we have 2 systems, MagX Starlet and MagX Star, for nucleic acid extraction. For the RNA extraction and preparation for COVID tests. And then a second system that is for qPCR setup, called the PCR Prep Starlet, and the combination of these 2 systems, so nucleic acid extraction as a first step on a Starlet, and the PCR Prep, PCR Prep on the second Starlet, enable the customer, the end user, to really speed up the process and to have an higher throughput.
Then we have 2 more questions. One anonymous again. How do you see the label-free detection is impacting both immune assay development and the existing immune assay market?
Label-free detection, yes, it's will impact, yes, the immunoassay development and also the market, the existing market. This is something that we are exploring as well, but it's something preliminary, let's say, to talk about. Sorry.
Okay. Then the last question comes from Kevin Miller. In the reseller agreement, does Hamilton continue to sell those instruments directly?
Let's say depends on the customer. There are some situations where the customer is asking for not a reagent rental conditions, so they wanted to buy directly from Hamilton and they want to buy the reagents from the kit provider. So in these cases, we have to, we discuss with the reseller and we follow the customer requirements at the end. So in this case, is the customer leading the, let's say, the negotiation between the parties.
I got just another question coming in from Mohammed Rafiq. He's asking, MagX and PCR Starlet, which are the kits validated in these platforms?
For the MagX, we have several nucleic acid extraction kits. For example, we have a kit from Thermo Fisher, the MagMax. We have the Omega Biotech kit. We have the Mackernagel pathogen kit. We have the Molgene kit. Yeah, these are the principal kits that we developed, we implemented, and the Promega, I think, and the Zyme also. We implemented the method on the MagX Starlet. For the PCR Prep Starlet, depends mostly on the customer. This is quite more heterogeneous, so there could be different kits coming from different manufacturer.
Seems like there are no more questions coming in for the moment. I hope that we've been able to answer all the questions. And as we had earlier mentioned to all our participants, please feel free to contact both Gabriela and Federico. Their email addresses are at the bottom of the last slide you're seeing at the moment. We'll be very happy to hear from you, of course. And well, it's almost 5 p.m. and that means we have another 5 minutes to end our webinar. And if there are no more questions, then I would suggest that.
Yeah, thank you to everybody for your participation.
Yeah, thank you. And please don't hesitate to contact us.
Thank you.
Thank you. Have a nice afternoon.
Bye-bye.